In this session, we will see about tetracycline expression systems. Hope you all know about tetracycline antibiotic. You know, tetracycline is an antibiotic which interferes with protein synthesis. This tetracycline will combine with the 16S ribosomal RNA of the 30S subunit of bacterial ribosomes. And this tetracycline is very effectively used against gram-negative bacteria. Since the discovery of the first antibiotic, that is penicillin, there is a tug of war going on between this antibiotic and the bacteria against which these antibiotics are being used. For a particular time, this antibiotic may win the race. That is, it can effectively inhibit the growth of the bacteria. But say after a particular period, now this bacteria will gain resistance against this antibiotic. Then the researchers, they will modify the antibiotic, then it will become effective against the bacteria, then slowly this bacteria will gain resistance against this antibiotic, and this process going on continuously. And this story is same with the case of tetracycline also. These bacteria, they have several mechanisms to defend this antibiotic, and the most common one and the very efficient mechanism is by simply pumping the antibiotic out of the bacterial cell. And for that, this bacteria use efflux pumps. They are nothing but membrane proteins present in the cell membrane of this bacteria, and this protein, it is coded by a gene designated as TET-A. And this tetracycline resistance, it is mainly by a tetracycline responsive repressor. This tetracycline responsive repressor is actually regulating the mechanism, and this tetracycline responsive repressor, or simply the repressor, it is coded by another gene called TET-R. And these tetracycline resistance associated genes, they are organized into an operon. And I am sure you are all familiar with the operon concept. Let us see this operon mechanism that is taking place in the case of this tetracycline resistance. This is the diagram of a general operon. Here you can see an operator, then a promoter, and the associated genes. And here you can see that this operator and promoter regions in the DNA, they are overlapping. And downstream the promoter, you can see the associated genes. And here I have included only the tetracycline A gene. You can have the tetracycline repressor gene also. Here you can see the repressor, and in the case of this operon, this tetracycline repressor, it binds with this operator, and when the repressor is found associated with the operator, this RNA polymerase cannot bind with this promoter, and as a result, transcription of the downstream genes are inhibited. This is happening in the case of any operon. Let us take another instance where there is tetracycline in the cell. We have provided tetracycline. Then you can see the repressor here. Along with that, you can see the tetracycline molecules also. And this tetracycline, it is having a high affinity towards the tetracycline receptor. There are specific sites in the repressor for binding with the tetracycline, and this tetracycline will go and bind with the repressor. Once it is bound to the repressor, a conformational change will take place in the repressor. And as a result, this tetracycline repressor, it cannot bind at the operator. That means the RNA polymerase can very well bind with the promoter and it will initiate transcription of the downstream genes. This is what is happening in the case of this tetracycline operon. And this mechanism, it gained importance in rDNA technology. It gained the attention of the researchers, which are very keen in searching for various mechanisms to regulate the expression of genes. Thereby, they can study the main activity, main function of a gene in a cell. And that gave birth to tetracycline expression systems. An expression system is something, it's a module with which we can regulate the expression of our gene of interest. We can upregulate, we can downregulate, we can say inhibit, or we can switch on the gene. Various possibilities are there with the expression systems. And tetracycline expression system is a very important tool in recombinant DNA technology or genetic engineering. And for constructing the tetracycline expression systems, what the researchers have done was they have created a fusion product. And in the fusion product, one component was this repressor. Then they have fused a transcription activator protein with this repressor to produce a tetracycline transactivator protein. And the entire mechanism of this tetracycline expression system, it is based on this fusion product, that is the tetracycline transactivator protein. It is nothing but a repressor taken from this tetracycline operon, then a transcription activator, and this transcription activator molecules, they are required for initiating transcription from a minimal promoter. Minimal promoter, they are similar to normal promoters, but they require this transcription activator proteins to initiate transcription from these promoters. So they have joined these two proteins to form a fusion product called tetracycline transactivator protein. And this is the module required for any tetracycline expression system. Here you can see a tetracycline response element is constructed, and it consists of a tandem array of tetracycline operons. Here you can see 7 tetracycline operators. Downstream of these series of operators, you can have the minimal promoter. And these tetracycline operators, they are same operator that is functioning in the normal tetracycline operon. So here, this is the fusion product, that is the tetracycline trans-activator protein. It has the repressor part. Then along with that, it has the transcription activator protein also. So it is a fusion product, and this repressor part of this fusion product has the ability to bind with this operator of this tetracycline response element. Similarly, all the other tetracycline operators are bound with the same type of fusion product. So here, when this fusion product is bound with the tetracycline operator, the transcription activators are also in place. That means they can initiate transcription from the minimal promoter. So here, the transcription from the minimal promoter will take place only in the presence of this fusion product at the tetracycline operons. Now, the researchers, they have created 2 types of tetracycline expression systems. One is the tetracycline off system. Let us see how this tetracycline off system is working. So here you can see the tetracycline response element. Then a downstream gene is there. It can be any gene. We can have any gene of our interest. So here these operators, they are bound with the fusion product. Then here comes our RNA polymerase. It binds with the minimal promoter. Remember, the binding or the transcription from the minimal promoter requires this transcription activators. As long as this fusion product is bound at the tetracycline operators, the minimal promoter will be active. That means transcription can be initiated from the minimal promoter. So the downstream gene is transcribed and the mRNA is formed. This is the normal thing that is happening in the cell. Now here, this gene, that is the downstream gene, whatever gene we have inserted there, that gene is active always. That means it is transcribing continuously. Then let us take another case. Here we are giving tetracycline to that cell. The same cell we are giving tetracycline. Now this tetracycline will go and bind with the repressor part of the fusion product. That means that conformation of that repressor is changed, and now this repressor cannot bind at the operator because its conformation is changed, and that change in the conformation is due to binding of tetracycline. So in the presence of tetracycline, that fusion product cannot bind at the operator. So no transcription will take place. So by providing tetracycline, you can switch off a gene which was normally transcribing. That is why this system is known as TET-off system. Here the gene was in an on condition, and by providing or by giving tetracycline, you can switch off that gene. Then coming to the next strategy, that is TET-on system. Here from the name itself, you can figure out what is going to happen here. Here the gene will be in an on condition, and you can switch off that gene by simply giving this tetracycline. Okay, let us see. So here you can have the tetracycline response element, then minimal promoter, and the downstream gene. Here that fusion product, that tetracycline activator, it is not that normal one. It is a mutated one. Here, this tetracycline transactivator protein is not the normal one. That tetracycline transactivator normal protein was able to bind with the tetracycline operator normally. But here, that fusion product is not able to bind at the operator in its normal condition. That means it is mutated. Mutated in the sense that conformation is slightly changed. You can see here the conformation is different. So it cannot bind at the tetracycline operator. That means the minimal promoter is not active. So no transcription will take place here. That means that gene downstream, the minimal promoter, is in a switch off condition. Now we are going to give tetracycline to this expression system. See what is going to happen. This tetracycline, as usual, will go and bind with the repressor part of the fusion product, and see the conformation has changed. So here, by binding with the tetracycline, that repressor part of the fusion product, it changes its conformation so that it can bind at the operator. Similarly, all other operators are bound with tetracycline, that fusion product. Now the minimal promoter is active so that the RNA polymerase can initiate transcription from the minimal promoter. So here that gene, it was normally in a switched off condition. By providing tetracycline, you can initiate transcription from that gene. So here we have two different strategies. One is used for switching off a gene, whereas the other one is used for switching on a particular gene. So using tetracycline expression systems, you can very effectively regulate the expression of the gene of our interest. So that's all for now. I think you are now very thorough with tetracycline expression systems. Thank you all. We will meet again in another session. Goodbye.